everyone, my name is Zitian Wang, and I am a PhD student from Dr. Pavi Palkana's lab at UCLA. Today, I'm happy to talk about our research on evaluating differential analysis methods for detecting robust cell states under the Milo framework, originally developed by Dan et al. Milo by Dan et al. Constructs cellular neighborhoods to elucidate the condition-specific cellular heterogeneity from single-cell RNA-seq datasets. To detect differentially abundant cellular neighborhoods, or DAs, the original Milo by Dan et al. Adopts HR. However, as HR does not remove outliers for differential testing, some significant DAs detected by Milo-HR are driven by very few outlier samples. To address this issue, we implemented DESeq2, which applies Cook's distance to detect outliers for testing DA under the Milo framework. We hypothesized that Milo-DESeq2 could detect more robust condition-specific cell states than Milo-HR because Milo-DESeq2 can remove DAs driven by very few samples. Now, let's look at the pipeline of Milo-DESeq2. Milo-DESeq2 uses the same soft k-nearest neighbor algorithm from the original Milo-HR by Dan et al. Could construct cellular neighborhoods for the input single-cell RNA-seq dataset. Then, Milo-DESeq2 generates the neighborhood cell count matrix where each row corresponds to the neighborhoods and the column corresponds to the samples. Each element in the matrix represents the cell number from the sample J in the neighborhood I. We assume the neighborhood cell count matrix follows a negative binomial distribution. Next, Milo-DESeq2 applies the likelihood ratio test to identify the condition-specific DAs. Finally, Milo-DESeq2 will calculate the Cook's distance for each cell count and removes DAs with outliers, which is the DAs that contain cell counts with very large Cook's distance. In this study, we utilized the three different single-cell RNA-seq datasets. The first dataset is a liver dataset from Ramachajan et al., containing 5 healthy individuals and 5 individuals with liver cirrhosis. This dataset was also evaluated in the original Milo paper from Dan et al. The second dataset is a dilated cardiomyopathy dataset from Cohen et al., containing 18 individuals with DCM and 27 healthy individuals. Lastly, we also included an independent DCM dataset from Chaffin et al., containing 20 individuals with DCM and 30 healthy individuals. To evaluate Milo-HR and Milo-DESeq2, we designed 2 analyses. For the permutation analysis, in each permutation, we randomly shuffled the sample identity of each cell and reconstruct the neighborhood cell count matrix and test the DAs with both methods. For the replication analysis, we first utilized the 2 methods to identify the significant DCM DAs from the Cohen et al. Dataset and calculated how much the differentially expressed genes from each method's significant DCM DAs replicated in the independent Chaffin et al. Dataset. Our goal is to demonstrate that the condition-specific DAs detected by Milo-DESeq2 are more robust than that from Milo-HR. Next, let me go over our results. First, in the liver dataset from Ramachajan et al., we observed that Milo-DESeq2 is more conservative than the original Milo-HR in detecting DAs because of removing DAs with outliers. However, Milo-DESeq2 is still capable of replicating all of the predicted enrichments in different cell types from the original Ramachajan et al. Paper. In addition, Milo-DESeq2 could successfully replicate the enrichment of cholangiocytes 2 and 3 in healthy samples, which was mentioned as one of the new discoveries from Milo by the Milo paper from Dan et al. Second, from our permutation analysis, Milo-DESeq2 did not detect any DAs during the permutations for both the Ramachajan et al. Liver dataset and the Cohen et al. DCM dataset, while Milo-HR detected DAs for 2.3% and 5.4% of all the permutations correspondingly for each dataset. Third, for our replication analysis, Milo-DESeq2 achieved much higher replication rate in the identified DE genes than Milo-HR in DCM-DAs for both endothelial cells and fibroblasts across 2 different DCM datasets. In conclusion, these results demonstrate that Milo-DESeq2 detects more robust condition-specific cell states with higher accuracy and replication rate than Milo-HR. Thank you very much for listening.